Hi everyone. So today we will be presenting magnetic biopolymers. My name is Bhagya.
My name is Danny.
My name is Martin.
My name is Angela.
And my name is Kaylee.
So before in this experiment, we'll mainly use three kinds of materials. So let me introduce to it. The first one is biopolymers. So if I want to speak about biopolymers, so I need to introduce polymers first. So what is polymer? Poly means many. So polymer is a kind of substance that made up of large quantity of repeating units. So we call these repeating units monomers. Then what is biopolymer? Biopolymer has two additional characteristics upon the polymers, which is they are produced by biological systems and they're biodegradable. So as we can see, and from the picture is DNA. So DNA is a biopolymer and is made up of nucleotide monomer, so which has 4 base pair ATCG. And the next one is ferrofluid. Ferrofluid is a kind of nanoparticles which is magnetic that's suspended in water, so which makes it have magnetism. And the last one is gel. The gel is solid jelly-like material, and it is kind of substantially dilute cross-link system. So let's take an example. I'll take sodium polyalkylate as an example. It often appears in diapers. So we all know that sodium polyalkylate absorbs water. So how does it absorb water to forms a gel? It is because when it dissolves into water, and the sodium ions will dissipate it and will come out from the polyalkylate. And at this time, there will only be negative ions in it. And because they have the same charges and they will repel each other and they extend to form like a network structure. And this will cause osmotic pressure. So the water will come in it, which makes it absorbs more water to form a gel.
So moving on to why magnetic biopolymers in specific. So biopolymers can be mixed with its cross-linking agent, so it can form a gel that way. So we can insert a ferrofluid magnet, as Danny explained, into the gel to give it magnetic properties. So the magnetic field will basically remain in wherever we're injecting the gel to apply to, and it will have a magnetic field that will keep it in place. So basically the magnetic field in the gel will keep it so that it doesn't move while it's applied to whatever application you would like it to go into. And throughout our research, we focused on something called osteoarthritis, which I will explain later, but it is we're basically trying to improve the weakened bone with more collagen, which is a protein, and has shock absorbent features, which the ferrofluid magnet provides it with. And we will also be going into various applications that magnetic biopolymers are also used in. So our objective is to create a magnetic biopolymer that has a gel consistency, so it will have a balance between solid and liquid, so it's still movable, and to have an active magnetic field, so it still has the magnetic properties given by the ferrofluids, and it would be used to relieve joint pain. So going into the medicinal applications, osteoarthritis is one of the main things that we wanted to focus on, and this is a common form of arthritis that is affected by millions of people worldwide, and it occurs when the protective cartilage that covers the ends of your bones wears down over time. And 2 other diseases and syndromes that are also related to this includes pataformal syndrome, and that's when it contains the disease describes the pain in which the front of the knee is like corroded by the bone weakens in front of the knee, and also known as a patella. And the last one is fibromyalgia, which is a disorder characterized by the widespread of musculoskeletal pain.
So some other medicinal applications using magnetic gel could be to release drugs by using the strength from the magnetic field. Another possibility could be to unclog arteries using the magnetic gel. And although there are not many applications for the magnetic gel in besides the medical field, it could be possibly used to prevent metals into the food or the removal of pesticides. And since magnetic particles do not interact well with sound waves, The magnetic gel could be used to create a noise-canceling earplug, which could be turned on and off in the presence of a magnetic field.
The biopolymers we used in this lab are iodocarrageenan, gelatin, and collagen. Iodocarrageenan is a polysaccharide polymer that comes from red algae. It has monomers of galactose and anhydrogalactose. It forms a structure by forming bonds of calcium ions, and the calcium ions bond with the sulfate groups. We also use gelatin, which is a protein biopolymer. Protein is divided by collagen and is prepared by the thermal denaturation of collagen. It forms a structure of hydrogen bonds. We also use collagen, which is the protein found in bones. It is a component of cartilage.
So focusing on the application of osteoarthritis, the arthritis that we wanted to focus on was in the joint, so that has cartilage that's affected, and cartilage is mostly consisted of collagen, which is a protein that promotes self-proliferation, and cartilage is a tough and flexible tissue, and it's also shock absorbance, which is why the ferrofluid magnets have a property that has shock absorbance as well. And basically, we wanted to enhance the bone functionality within the affected area, so the gel would provide the shock absorbance with correlating with the ferrofluid magnet as well. And this serves as an alternative for implants because implants have a background of causing infection. So this is a positive alternative for that.
To create our ferrofluid, we needed to create nanoparticles. Usually for microparticles that have multiple domains. Domains are areas where the magnetic field of atoms align with each other. But as you go smaller into nanoparticles, eventually there's only one domain, which results in superparamagnetism. This makes them very responding to a magnetic field. We also use magnetic nanoparticles also make the substance they're in magnetic, and we had to alter our nanoparticles to be compatible with water. We use 3-mercaptopropanoic acid, and this is a capping ligand we use to cover the nanoparticle, and this is a polar tail, which is able to be suspended in water.
So after doing all of our research, we decided that our overarching question for our project would be, which mixture of biopolymer and ferrofluid can create a magnetic and movable gel? And our original hypothesis was that if 2 milliliters of ferrofluids, if we mix 2 milliliters of ferrofluids into 25% collagen, then a magnetic gel will be created, which can later be injected into a pain to decrease into the joint.
So the first part of our procedures was to find the appropriate gel consistency. So we created gels with different concentrations of collagen, iodocarrageenan, and gelatin. And we measured the Young's modulus for these, which is basically the elasticity or the resistance to compression. And we did that by stacking pennies on these gels. And so we basically wanted to find a biopolymer gel that is movable, so we want a balance between a liquid and a gel consistency.
The next part, we have to make our ferrofluid solution. We use ferric and ferrous chloride as our iron sources and ammonium hydroxide as our oxygen source to make iron oxide. And then we use 3-mercaptopropanoic acid to cap it off, to stop it from growing further. We mix about 1 milliliter of each solution to biopolymer gels.
So next, we tested whether the ferrofluid works in our gels. So we first measured the Young's modulus for each gel we made, and then we recorded data and plot a graph. After that, we tested the application specific, which we put in magnet, placed our ferrogel onto glass slide and move it with a magnet to see whether the ferrofluid works in our gel.
So putting this into a context of osteoarthritis, so someone would inject this gel into the joint or the location that it would be necessary in, and it would basically, we would use another magnet to move it around to fit the person's knee or joint. So that's where we wanted to test the movability with a magnet.
So we mentioned Young's modulus several times before. So let me introduce what is Young's modulus. So basically, Young's modulus is also called elasticity modulus. So it basically measures the elasticity and the stiffness of a substance. So the principle of Young's modulus is basically because when a force exerted on an object, it will cause deformation. So a more elastic object is, so more deformation it has. So Young's modulus basically focuses on a relationship between the stress and the strain, these two kind of things. So it has an equation, which is the force exerted multiplied by the original length divided by the product of the contact area and the increasing in length. So, but that's a general equation of Young's modulus. So we made a more specific equation for our experiment, which is, so we use this equation to do some specific calculation in our experiment, which is the number of pennies and multiplied by the mass of each penny and the gravity constant, also multiplied by the thickness of gel divided by the volume of the penny to calculate the Young's modulus for each gel we made.
First, we chose collagen as our first biopolymer because collagen is the most commonly found protein in bones. In addition, collagen helps cell proliferation and helps bind to other proteins. However, through our experiment, our collagen gels would not be able to turn into gel consistency, so we decided to use other biopolymers such as iota-carrageenan and gelatin, which is a derivative of collagen. So here are pictures of our collagen gels. In the upper right-hand corner, that is our 20% collagen gel. And next we made our 25% collagen gel. And then finally we decided to spike up our collagen by making a 50% collagen in order to try to make a gel, which still did not work in the end.
So moving on to the second biopolymer that we tested the one that we did previously last week, which was iota-carrageenan reacting with the calcium chloride. So calcium chloride is actually the cross-linking agent for iota-carrageenan, and we did 4 types of concentrations for the calcium chloride. So basically, we would have a constant for the iota-carrageenan, which was 1%, and then we had a variation of the CaCl2 concentration. So we would heat up the entire solution and then we would cool it, and that's how the gel would be created. So we had 3 variables for the concentrations that we had. So first was just ferrofluid. So we did that for every concentration of CaCl2, and then we just had one with 25% of collagen, and then we had one where combined both of them. And highlighted, you could see the ones that we found most ideal, which had the balance between the solid and liquid state of matter, and it had slight magnetism. So as you can see here, these are all of our gels for iota-carrageenan and CaCl2, and here are the ones. This is the 0.25% of CaCl2, and this is the 0.3% of CaCl2, which we found most ideal.
As we tested our Young's modulus, we found the more CaCl2 we put in the gel, the higher the Young's modulus was.
So our third biopolymer is gelatin. So we made gelatin gels with 30% and 50% concentrations of gelatin, and we also added 1 milliliter of ferrofluid as well as collagen into the gels. And as you can see, all of our gelatin gels came out really stiff, and even the ones with ferrofluids in it were very slightly magnetic, so it wasn't as magnetic as we had hoped. So here are pictures of our gelatin gels. Those are 50% and 30% gelatin. And as you might be able to see, they're really stiff and really dry. So that wasn't the consistency that we were looking for. So far, our gels haven't really been magnetic either. Well, they have been, but not as much as we had hoped. So we decided to make 5% gelatin gels, and we just wanted to test the impact of the ferrofluids on those gels. So we added 0 drops, 15 drops, and 30 drops of ferrofluid into those gels. And even in the 30-drop gel solution, it was slightly magnetic, and it wasn't really movable. So we wanted to make it more magnetic.
When we tested the Young's modulus, we found that the 50% was very high, and This is the reason why it was so stiff.
So at this point, we haven't really got a magnetic gel, so we decided that we would just take one gel that was not too stiff but not too liquidy, and we would just spike it with a whole bunch of ferrofluids. So in our 0.1% CaCl2 and 1% iota carrageenan gel, we added around 2 milliliters of magnetic nanoparticles, and that made the gel extremely magnetic and movable.
As you see, it responds to magnetic fields, like we hope.
So in conclusion, as we saw through the graphs, the Young's modulus increases as the CaCl2 concentration increases as well, and gelatin, because as we added more of the concentration of the actual biopolymer, it added more of the liquid and solid substance that we were looking for and hoping to receive. And it was directly proportional, so as both of them increased, it both increased at the same time, so it was directly proportional. And this is until it reaches the maximum point of gel stiffness before it reverses the reaction and becomes a liquid once again. And the lower the Young's modulus, the more movable it was. So as you can all expect, a liquid is more movable than a solid. But we were trying to find the balance between a liquid and solid, which is why the younger the Young's modulus, the more movable it would be. And the higher the concentration of ferrofluids, the more magnetism it had, which is also kind of a given because the more magnets that you add, the more magnetic the field would be. And the elasticity of cartilage we found was 7.79 MPa for normal cartilage and 1.36 MPa for arthritis. So that's just like a comparison to what we actually found.
So after this experiment, we also came up with several refinements. The first is we noticed that the binding ligand we use is 3-mercaptopropionic acid, which is toxic and will do harm for human bodies. So since this kind of gel will be injected into human bodies, so we cannot use this. We can either make it non-toxic or we switch to another polar ligand. The second is we also should control the amount of ferrofluid in the kind of gel because also it should be injected into human bodies, so we should control the amount of ferrofluids in it. So that's all. Thank you.
Very nicely done. Quick question about the magnetic stuff inside the body. Are you concerned about the use of MRIs?
Yeah, so multiple questions have arisen as like a magnet and you're walking through airport security and it beeps. So what we found was just the doctor that's taking, or like the physician that's taking the MRI would be aware of the magnet that's in the body, and this is something that we'd still have to further research, but they would probably be aware of the magnet that's in the body, so that would yeah.
All right, I have a question. So you did mention at the end the modulus for cartilage, or the elasticity for cartilage. So how does that actually compare to the data that you got, and what sort of how would you further refine your material to match what it is you're looking for this application of cartilage in the knee?
So our experiment, we also calculated the Young's modulus for our experiment. So we can see that the normal elasticity of cartilage is 7.79 MPa for normal. So we need to make our gels which has a similar Young's modulus for that. So from our experiment, I see the iota carrageenan with with concentration of calcium chloride with 0.3% would be the most near the Young's modulus for cartilage.
And to further reiterate, so cartilage is mostly composed of collagen, which is not the protein that we tested and got our the data from this graph from. This was from iota carrageenan and CaCl2. So with the cartilage, it would be consisted of the collagen biopolymer and not this one.
Yeah, because see, so for that data, you're at like kilopascals, I think,
Yeah,
right?
yeah, MPa. So
Well, so I think that looks like kilopascals. Megapascals would be like 10 to the 6th. So it's actually like quite an order, like a few orders of magnitude higher of a modulus. So there'd probably be some sort of work you'd have to do to like figure out how to get it to really match cartilage if that's the application you are going for. But that might conflict with the sort of movability of the gel, right? Because if it's super, super stiff, that might be challenging. So can you think of some type of way of getting around that sort of conflicting problem of you need a stiffer gel, but you also want it to be a movable gel?
there's actually an application where one of the instructors actually told me about. So there's like a paper that consists like a prescription, like a drug inside of it. So when the magnetic field is attracted to it, when it's injected into the human body, it'll like release the medicine into the body. So with the magnetic field, it has that property of doing that. So maybe for further research, we could find out something that when the magnetic field is present, the gel could become stiffer in a sense, and when it's not, then it would be more movable.
Or thinking maybe of some way of like cross-linking it once it's inserted and it's already sort of in place, as opposed to having it already like pre-made or something like that. Other questions?
Hi. Have you guys seen any research on the general biocompatibility of the ferrofluid itself?
Yes, we have researched that some amounts are non-toxic, which we would have to also further research. But the amount is uncertain.
Okay, thank you.
All right, if there's no other questions, then let's thank our group. Nicely done.